Hello, all. So we'll just wait for 2 minutes. Let everybody join and then we'll start the session. So, is my voice clear? Just give a thumbs up if my voice is clear to you. Okay, fine. So, now we'll start with our session. Okay. Hmm. So, a very good evening. So, in today's class, we'll be dealing with the questions which were covered in the week 11 of our NPTEL online course. Yes. So after this, we have two more classes left. So in the next class, we'll do the remaining portion and the last class. So generally, it is a 12-week class. Okay. I mean, 12 online sessions. So in the next online session, we'll try to cover the questions which are covered in the next week 12th, and the last class, that is week 13th class, that would be a summarizing class. So in that class, what I'll do is that I'll try to quickly summarize the whole syllabus by solving some of the questions from each week. Okay, so I'll take questions from each week and we'll try to understand those questions and how to solve those questions, and in this way, we'll try to summarize whatever we have learned in this course up till now so far. And that would be the end of your course. So we will have, after this, we have two more classes left. And then obviously you have your exams. So the last class, that is your week 13 class, will actually help you to kind of revise everything. So I would request everybody, whoever will watch it offline on YouTube also, to please attend that last class, because that is help beneficial for you only. Okay, now coming to our today's solving session. So, as we were studying in the last class, so we started with the mammalian development. So, first we dealt with the Drosophila development. We dealt how to learn the anterior to posterior axis formation and dorsal ventral formation. Those things we have learned. We have also learned about stem cells, the basics, and we have also learned about some basic cell signaling or cell biology which is needed to study the developmental biology. And then we switched to plant developmental biology, and we studied in brief about the plant and then we studied in brief about the plants. And then in the last class we switched to the mammalian development, and in this week we'll be continuing the mammalian development questions. So the one for those who have just joined, Sangeeta, I was just telling that this is the class. After this, we'll have a class where we'll finish our course. So, in week 12th class, we are going to finish our course, and then we'll have an extra session before your examinations, week 13th session. The exact date and time would be notified to you through mails, where we'll try to summarize everything. Okay, that will be like a revision class for you all. So, if you have any questions, you can ask those questions as well in that class. Okay, now we'll start with the questions for today. So the first question is, so as we'll do the questions, we'll try to understand. Hmm. Uh, I request Ajay to please mute yourself. Yeah, thank you, Ajay. Okay, so now we'll start the class. So our first question is, during gastrulation, the cells lose A. Encadherin, B. E-cadherin, C. P-cadherin, and D. NP-cadherin. So these are your four options. I'll give you 2 minutes. After 2 minutes, I will switch the screen. So whatever answers you think, please write it in the message box. So your time starts now. It's an easy one only. One more minute left. After 1 minute, I'll switch the screen again. Okay, so your time is up now. Now I'm going to switch the screen. Fine. So let us see what you have answered. Okay, so Ajay has answered B and Jasrina has also answered B. And the correct option is B, that is E. Catherine. Good. Ajay and Jasrina. So let us see what the question says. Okay, so during gastrulation, the cells lose N-cadherin, E-cadherin, or P-cadherin. So basically, you need to understand that what happens during the gastrulation process. So during the gastrulation process, one very important event take place which will decide what kind of cells would be there. And what is that event? That event is EMT. What does EMT stands for? It stands for epithelial to mesenchymal transition. So during gastrulation, epithelial to mesenchymal transition takes place. And during this transition, the cells they lose E-cadherin because it is a protein which is involved in adhesion, and this allows the cells to migrate and differentiate into various tissues. Okay, now you should understand that cadherins in general, cadherins are involved in cell to cell adhesion. That means the joining of two cells with each other, the adhesive property is facilitated with the help of cadherins. And there are three major types of cadherins which are involved. Okay, there are three major types of cadherins. The first one is your E-cadherin, then you have N-cadherin, and then you have P-cadherin. Okay, your E-cadherin stands for epithelial. This is your epithelial cadherin. Your N-cadherin, N stands for neural, and your P, here your P stands for placental cadherin. Now, an E-cadherin, as the name suggests, it is an epithelial cadherin. That means it is majorly found on simple epithelium tissues, simple squamous epithelium tissues, which are somewhat cuboidal in shape. Okay. Now, when epithelial to mesenchymal transition take place, these epithelial cells, this is not a perfect cuboid, I'm just telling you that something, these epithelial cells, because now the transition has occurred, they change to mesenchymal cells, which are spindle shaped. Okay. Now, why this happens? Because these epithelial cells are very much stick to their places. They do not move. But these spindle cells can easily move. I mean, they have a property of migration. And this movement is facilitated with the help of cadherins. So, your E-cadherins, which are present in the epithelial cells, they keep the cells together with each other. They keep it intact. They does not allow it to let loose. But when the transition takes place, when epithelial cells starts turning to the mesenchymal cells, then your E-cadherin changes to N-cadherin. N-cadherins are mostly present in neural tissues, and also it is present on mesenchymal cells. So what happens now, since N-cadherins are there, the mesenchymal cells have formed. Now they have let loose to themselves so that they can migrate. So this epithelial to mesenchymal transition is also accompanied by the change in the expression of the type of cadherin. In epithelial cells, E-cadherins are expressed, but when the transition takes place, then N-cadherins are mostly expressed, which are involved in mesenchymal tissues. And your P-cadherins are the placental cadherin, which are mostly present in placenta and in some basal epithelium also. Now, always remember that N to E transition, sorry, E to N transition takes place during epithelial to mesenchymal transition, and this epithelial to mesenchymal transition event takes place during gastrulation. Also remember that epithelium to mesenchymal transition is a general event. That means it is not only observed during gastrulation, it is observed in other stages and in other phenomena in human body as well. One very important phenomena in which this epithelial to mesenchymal transition is observed is cancer. So, in the case of cancer, this EMT takes place, and therefore, it is one of the very important reasons that why cell becomes tumorous. Also, during pregnancy, the uterus also prepares itself for the incoming blastocyst. The blastocyst, the fertilized egg, it has to come through the oviduct to attach itself to the uterus, right? So, before the uterus comes, sorry, the before the blastocyst comes, the uterus also prepares itself. So during its preparation process, again this epithelium to mesenchymal transition event take place. And in every case, like in each and every case, your E to N transition also take place. So this is important. I hope this is clear to you. Okay, this epithelium to mesenchymal, if for yeah. So this epithelium to mesenchymal event is important. Fine. Now we'll come to the next question. The question is, structures that gives rise to blood vessels of the umbilical cord. What are those structures called? A. Extraembryonic mesoderm. B. Embryonic mesoderm. C. Endometrium. And D. Chorion. So these are your four options. It's 7:12. I'm giving you 2 minutes to solve this question. At 7:14, I will switch the screen. Whatever your correct answers, whatever you think are the answers, you can write in the message box, and please try to attempt. Do not feel that what if my answer is wrong. It's fine. You're just learning. These questions, whatever we are dealing here, they are not going to be assessed. Okay? It is just for your practice. So I would request everybody to at least try and attempt. Whether we'll see whether it is correct or not, or anyways I'm going to explain the concept of each and every question. I always explain that. Okay, try to solve this question. 1 minute left more. Okay, now I'm switching the screen. Hmm. So what you have answered? Okay, so Ajay has answered A and Amla has answered B. Okay, fine. A is extra embryonic mesoderm and embryonic. Okay, the correct answer is A, that is extra embryonic mesoderm. Fine. This is the correct answer. And in this one, this is the correct answer. Okay. Now let us try to understand this question. So the question says structures that give rise to the blood vessels of the umbilical cord. So blood umbilical cord, the blood vessels of the umbilical cord, they are derived from the extra embryonic layer of the mesoderm. Okay, and this extra embryonic layer of the mesoderm and this umbilical cord, this forms a vasculature which connects the embryo to the placenta, which helps in the exchange of nutrients and gases between the fetus and the mother. So just you have, this is not a very conceptual question, it's a direct question. Just you have to understand that this umbilical cord is very much important because it is a point of connection between the embryo and your fetus. Okay, and it helps in the exchange of nutrients and gases between the fetus and the mother. So the embryo is connected with the placenta with the help of this umbilical cord, and this umbilical cord is derived from your extra embryonic mesoderm. Okay, just a minute, sorry. Yeah, now the next question, it's an interesting question. I'll just do like this. Yeah. Okay. The next question is, if we separate the two cells of a two cell embryo, the generated extra embryonic structures will be A, one chorion and one amnion, B, one chorion and two amnion, C, two chorions and two amnions, and D, no extra embryonic structures. So these are your four options. 2 minutes to solve this question. I'll switch the screen at 7:19. It's 7:17 right now. Okay. So try to solve these questions. Question. Think carefully what the question is asking. One minute more. Okay. Now time is up. Hmm. What you have answered? Hmm. So, okay. Jasrina has answered C, Amla has answered D, and Ajay has also answered C. Let us see. Okay, so the correct answer is C. That is two chorions and two amnions. Now, can anybody tell me the difference between chorion and amnion? What is the difference between chorion and amnion? Can anybody tell me the difference? Hmm? Does anybody knows the difference between chorion and amnion? Chorion is blood vessel, okay, and amnion, amnion is fluid. Okay, that way also if you are saying it's fine, but understanding with respect to the embryo, chorion is basically the outermost membrane that surrounds the embryo, and ultimately it develops to the fetal part of the placenta. Okay, so you have to understand with respect to your embryo when I'm talking about. So chorion is the outer layer of the amniotic sac, and amnion is the inner layer of the amniotic sac. What I'm saying? I am saying that chorion is the outer layer, outer layer of the amniotic sac, and amnion, it is the inner layer of the amniotic sac. Another difference is that since it is the outer layer, it is thicker and tougher, and since it is an inner layer, it is more thinner and it is elastic. So this one is a little thicker and a tougher layer, and this is a little more thinner layer. Okay, it is more elastic also. Now that we know what is the meaning of chorion and embryon, let us try to understand this question. Here they are saying that if we separate the two cells of a two-cell embryo, the generated extra embryonic structures will be what? So, if the two cells of a two-cell embryo are separated, then each cell can develop into an individual embryo. Remember, two cells are there of a two cell embryo because it is at the two cell stage, it is very much totipotent. So each cell can develop into an individual embryo. So if each cell is developing into an individual embryo, that means this will result in the formation of two separate extra embryonic structures. Okay, that means each embryo will develop its own chorion and its own amnion. Therefore there would be two chorion and two amnion. So what I'm trying to say here is that each embryo, see, each embryo has one chorion and one amnion. Okay. And now since it is a two cell embryo, we have a two cell embryo. So here each cell will develop into a separate embryo. So there is one embryo, there is another embryo. So this embryo will have its one chorion and one amnion, and this embryo will also have one chorion and one amnion. So total how many chorions will be there? Two chorions. And how many amnions would be there? Two amnions. So I hope this question is clear to you. Okay. Now Next question is, freshwater stickleback fishes lack dash because dash enhancer is not active. First, dorsal spine, PICS 1. Second is pelvic spine and PICS 2. Third is pelvic spine and just a minute. PITS 2. And third is dorsal spine and PITS 2. So you have 2 minutes to answer this question. It is 7:24 right now. I will switch the screen in 7:28. Okay, sorry, 7:26. 2 minutes. One more minute left. Okay, your time is up. Hmm. Let us see what you have written. Ajay, it is not totipotent, just pluripotent. No, Ajay, you are wrong. Amla, you are right. It is totipotent, it is not pluripotent. Okay, you have to understand the difference between totipotent and pluripotent. Just tell me, can a pluripotent cell give rise to whole embryo? No, pluripotent cell cannot give rise to a whole embryo. Only a totipotent cell can give rise to whole embryo, not a pluripotent cell. So if you are saying that C is the correct answer, okay, C. C, this was the option. Yeah, that two chorions and two amnions will be formed. That means that these two cells have the property of differentiating into a complete organism. Okay, the two cells of that two celled embryo, each cell has the property of developing into a complete organism. That itself says that they are totipotent, they are not pluripotent. So clear your concept, Ajay. Up till fourth cell stage, the embryo is totipotent only. It is not pluripotent. Pluripotent embryo cannot give rise to a whole new organism. Pluripotent cells cannot give rise to a whole organism. Only totipotent cells can give rise to whole organism. So there is a difference between totipotent and pluripotent. Initially, when the after the fertilization has taken place, the initial stages are totipotent, and then later on they come to pluripotent stage. And if the question says that they have, if the explanation and the you yourself has answered that each cell is giving rise to a whole embryo, so this pluripotent cells cannot do. Okay, they cannot give rise to whole organism. Only a totipotent cells can give rise to whole organism. So I think, Amla, you are correct that early embryonic cells are totipotent in nature. They are not pluripotent. Up till fourth cell stage they are totipotent. Okay, you should always, if you don't have the if you have any confusion, I would request everybody to go and just read about the difference between totipotent, pluripotent, multipotent, unipotent, and like whatever the potencies are in stem cells, because these are the basics. And I can understand because generally people confuse between totipotent and pluripotent. Okay. So what property does totipotent has that pluripotent does not have? So pluripotent stem cells can differentiate into most or all cell types, but the extra embryonic layers, the extra embryonic tissue, ectoderm, endoderm, and mesoderm, those cannot be formed by pluripotent. But your totipotent cells can form embryonic as well as your extra embryonic tissues. Okay, so totipotent cells have the highest differentiated potential, and it can differentiate into each and every cell and can make a whole complete organism. Whereas pluripotent stem cells cannot give rise to a complete organism. Okay, now we'll come to, I think this confusion is clear now. Now we'll come to the this question answer. So what Amla has answered 2, and Jasrina has also answered 2. Ajay, totipotent can rise to trophoblast. Yes, so see, you have written, no Ajay, that totipotent can give rise to trophoblast and every cell, pluripotent except trophoblast. That means that the cells are totipotent because each cell is forming a complete organism. That means, or embryonic as well as extra embryonic layers, everything is formed. So inner cell mass is your embryonic layers. Trophoblast, trophectoderm, which forms your ectoderm, endoderm, mesoderm, those are your extra embryonic. Okay. Now, so all have answered B. Let us see what is the B option. B is pelvic spine and which one? It is the correct answer. Okay. Now, let us try to analyze this question. The question says that freshwater stickleback fishes, they lack dash because of dash enhancer is not active. So, they lack pelvic spine. They lack pelvic spine because PIX1 is not active. So, what is, first of all, you should know what are these stickleback fishes. So, basically, they are your small elongated fishes like finned fishes, ray finned fishes. Okay, and why they lack a pelvic spine? Because the gene that is PIX1, its enhancer, which is involved in the development of pelvic structures, is not active. And this is an example of evolutionary adaptation where changes in the regulatory elements, for example, enhancers, can lead to morphological differences between populations. And this is a very important point. I would like to again focus. I always try to emphasize on this point in whenever this kind of topics come, that all the genes are present in each and every cells of the body. I think I have explained this point a number of times, that genes which are responsible for your eye colors are also present in your lungs, and genes which are responsible for your lungs are also present in your eyes. But why the genes are not expressing in other places? Because of their differential gene expression. So the genes are expressed in a cell in a and a temporal, I mean, origin and temporal manner. That means cell specific manner expression, that genes which has to express in the eyes will not express itself in the lungs, although they are present in the lungs. So it's not only about the presence of a gene, but it's more about the expression of the genes. Now, how this differential gene expression is regulated, we have already studied in our previous classes. One of the key factors which controls the expression of genes, or rather the differential expression of genes, are the regulatory elements like the repressors, enhancers, promoters. So, these are some of the regulatory elements which are present within the gene, within the locus, which helps in the expression. So in this case, your PIX1 gene is present, but why it is not getting expressed is because the enhancer which is involved in its expression is not active. Okay, so PIX1 would be active only when the enhancer which is involved in its expression is active. So the enhancer which is involved in its expression is inactive, and therefore PIX1 gene is not getting expressed. And because this PIX1 gene is not getting expressed, so pelvic spine formation is lacking. So you can see that changes in the regulatory elements, it can lead to morphological differences. Okay, this line is very important. What I'm trying to say, that changes in regulatory elements can lead to morphological differences. And what are the regulatory element that we're talking about here are enhancers. We have promoters also. We have silencers also. These are all your gene regulatory elements which regulates the expression of the genes, which helps in the differential expression of the genes. So I hope this point is clear. Now what Ajay has written, long paragraph. Okay, so whatever theory you have written, Ajay, I don't have time to read all these things. Just remember what I told others, that you can just read whatever theory he has written. These are totipotent cells, because if they are not totipotent cells, they cannot give rise to the whole the whole organism. Okay. So before this stage, any of the cells could have become trophoblast to inner cell mass. Therefore, these blastomeres are called totipotent because they can generate all cell types. While the inner cell must buried within this, while giving rise to all other cell types of the embryo, it does not produce trophoblast. As a result, they are not called totipotent. Yes, instead they are called pluripotent because they have the ability to make. Yes, see, whatever this Ajay has written, that is correct, but that it doesn't go with, it has nothing to do with the question that has been said. The pluripotent cells cannot give rise to trophoblast, it can only give rise to inner cell mass, and totipotent cells can give rise to both trophoblast and inner cell mass. And but these cells, two cell stage, they are totipotent in nature. Okay, now let us move to the next question. Hmm. A person is resistant to Plasmodium vivax. What might be the reason behind that? A. Mutation in HBA. B. Mutation in specific enhancer of Duffy. C. Mutation in PIX2. And D. Mutation in HBA1C. Okay, this is your question. And right now it is 7:37. 2 minutes to solve this question, and I will switch the screen at 7:39. Okay. 1 minute more. Okay, now I'm switching the screen. Okay, fine. Let us see what you have answered. Okay. Jasrina has answered B and Amla has answered first choice B, second choice C, but not really sure. Okay, fine. So let us see. The correct answer is B. So I think both have answered it correctly. Good. A person is resistant to Plasmodium vivax. What might be the reason behind it? So the correct answer is B. That is mutation in specific enhancer of DUFFY Duffy. Okay. So now let us try to understand this question. So Plasmodium vivax is the parasite which is responsible for a form of malaria which requires the Duffy antigen on the red blood cells to enter and to infect them. Okay, so this Plasmodium vivax will infect if it has the Duffy antigen to enter and to infect it. Now, people who have a mutation in this gene, that is your Duffy gene, and where the mutation should be, the mutation can be maybe at the antigen receptors of the Duffy gene or maybe at the enhancer region, that is a different thing. But simply you try to understand that people who have a mutation in this Duffy antigen gene, uh, so now what will happen? It will make the Duffy antigen non-functional and therefore the person will become resistant to Plasmodium vivax infection. So basically on red blood cells, this Duffy antigen, if there is the Duffy antigen is present, then Plasmodium vivax, Plasmodium vivax will come and it can infect it. Okay, but if there is any mutation in this Duffy antigen, then what will happen? Then this Plasmodium vivax cannot bind to this Duffy antigen because now this Duffy antigen has become non-functional. And since Plasmodium vivax cannot bind, and therefore those person which have a mutated Duffy antigen, they will become non-functional and they will become resistant to the Plasmodium vivax infection. Okay, so this is about it. So Duffy antigen is basically responsible is particularly required for the Plasmodium vivax to come and bind to it. So this option is correct. Now what is this PIX2? We have already studied just about it. It is associated with uh, contraction. Basically it's a gene which is associated with muscle contraction generally. And this HbA1c, this is basically a test for glucose level measurement. So I hope this question is clear. Okay, now we come to the next question. Hmm. Which of the following statements regarding mammalian development are false? First is ectodermal cells are the first to enter the embryo through the primitive streak. Second, allantois is a rudimentary organ which doesn't contribute much to mammalian embryonic development. C. Cell fate is specified as and when they entered the embryo via primitive groove. D. PCGs are among the last to migrate to their final location within the embryo. And E. Trophoblast cells are required for nutrient and waste exchange. Okay, so you have 3 minutes to solve this question, and right now it is 7:43. So at 7:46 I'll switch the screen. Try to read each and every this thing options and then try to answer it. Okay. 2 more minutes left. 1 more minute left. Okay. Now I'm switching the screen. Hmm. Okay. Fine. Let us see. Okay. So Jasrina has answered A and Nabla has answered A and B. Fine. See, so the correct is A is ectodermal cells are the first to enter and okay. So the correct answers are A. I mean ectodermal cells are the first to enter the embryo through the primitive stage. Then cell fate is specified as and when they enter the embryo via the primitive group, and trophoblast cells are required for nutrient and waste exchange. These are the false statement. Okay, so you have to mark the false statement, that is important. Okay, now see, we'll try to understand each and every point line by line. So the first point is ectodermal cells are the first to enter the embryo through the primitive streak. So is it a correct or wrong? It is wrong because mesodermal and endodermal cells are the first to migrate through the primitive streak and not the ectodermal cells. Okay, so this is a false statement because mesodermal and endodermal cells are the ones. So this is false. This is false statement. Okay. Then next is allantois is a rudimentary organ which doesn't contribute much to mammalian embryonic development. It is true or false? It is true. Why it is true? It is true. I mean, comparatively, you have to see whether it is true or not. Okay. It is not, we cannot say it is completely true, but in comparison to the other option, it is comparatively true to an extent because allantois in mammals is less significant compared to other species like birds. Okay. So, allantois is an important organism in other species. For example, in birds. Here, the question says that allantois is a rudimentary organ which doesn't contribute much to mammalian embryonic development. Here, we are specifically talking about mammalian embryonic development. So, in regard to mammalian or embryonic development, it is not completely true. I mean, it is not completely important. In birds, it is very much important. So, basically because it contributes to the early blood vessel formation in mammals and then it becomes the part of the umbilical cord. Okay, so comparatively you have to see, okay, this is not completely false also, but compared to the other statement it is true. Okay, sometimes these options are confusing, and because it is given as true, so this is the most probable explanation that I could get that why this statement was uh, given true in your answers. Okay. Then next is cell fate is specified as and when they enter the embryo via the primitive groove. Is it a correct statement? No, it is a false statement. Why? Because see, some initial fate decisions are influenced by migration through the primitive streak, and I agree to it. But cell fate specification is not fully determined at this point, and it remains little flexible. Okay, so there is not a complete cell fate specification. It still remains little flexible, and cell fate is specified gradually through signaling interactions and migrations also. So basically, the cell fate specification through signaling interactions after migrations, at that time also some cell fate specification happens. So initial fate decisions are influenced, but it is not complete, it is not fully determined. So therefore this option is also false. This is incorrect statement. Then primordial germ cells, that is PCGs, are among the last to migrate to their final location within the embryo. So, is it a true statement or a false statement? It is a true statement, because primordial germ cells, they migrate later to the developing gonad. So, this statement is correct. Then your last statement is trophoblast cells are required for nutrients and waste exchange. Now, according to the answers that has been given to you, it has been regarded as a false statement. The most logical explanation that I could think that why this is a false statement is that while trophoblast cells, they do facilitate the formation of placenta and supports the embryo implantation, they themselves do not directly handle the nutrient and waste exchange. Do you understand? They are themselves not handling this nutrient exchange and waste exchange. Basically, the placenta, which forms partially from the trophoblast, they handle these processes. So although placentas are all derived from trophoblast only, part of the placenta is derived from trophoblast only, but trophoblast cells are not directly handling this exchange. So therefore this becomes a false statement. So D is true and this is false. So these three options are your false statements. So this is the most the most logical explanation that I could think, that why we can say, how we can say that trophoblast cells are not required for nutrient and waste exchange. Although, see, it is all whenever you're solving these kinds of multiple choice questions, no, it is a lot uh, context dependent and comparative. So same statement can be true for for one case, it might be false for another case. For example, if we are writing a more generalized statement in one option, in one of the questions, where other statements are completely different from this generalized statement can be a true one. In another question where we have a generalized statement also and a more what to say, a more specific statement also, then more specific statement would become the correct one and this would become the wrong one. So same statement can become, here it was right, here it was wrong. So in these kinds of questions where you have like sentences and you have to choose the correct statements, you have not only to consider individually the sentence, but also try to compare it with other options. This is a trick. Do not consider a statement as an individual statement when you are solving such kinds of questions. Always compare that with respect to the other option, does it look more truer or not. Okay, so this is the way you have to solve these kinds of questions where you have options given to you. Fine. Okay, this is the next question. What does the question says? Yeah. Which of the following statements regarding mammalian development are true? First is gastrulation begins at the Hensen's node. B. Hypoblast cells will form the future endoderm of the embryo. C. Mesoderm cells originate in the epiblast and migrate through the primitive streak to enter the developing embryo. D. Exogenous expression of nodal may result in conjoined twins. And E. Hypoblast is responsible for positioning of the gastrulation site. So these are your statements, and you have to choose the correct statement. In the last one, you had to choose the false statements. Here, you have to choose the correct statement. So right now it is 7:54. I will give you 3 minutes. At 7:57, I will switch the screen and the time. Now it's 7:55. Okay, I will switch at 7:58 now. Okay. 2 more minutes left. Think of all the options and then try to solve. Try to analyze each option logically and conceptually. Hmm. Think. 1 more minute left. Okay, now I'm switching the screen. So, Somla has answered B and C. Fine. That is, hypoblast cells will form the future endoderm of the embryo, and mesodermal cells originate in the epiblast and migrate through the primitive streak to enter the developing embryo. Okay, fine. The correct answers is these ones. These are the true statements. Mesodermal cells originate in the epiblast and migrate through the primitive streak to enter the developing embryo. Then, exogenous expression of nodal may result in conjoined twins, and hypoblast is responsible for positioning of the gastrulation site. Now, let us try to understand each and every question. Okay, so first is gastrulation begins at the Hensen's node. First of all, you should know what is the meaning of Hensen's node. So, there are two terms. Okay, I'll just try to explain first, because see, whatever questions we are dealing, I always say that it doesn't matter whether we are solving 10 questions or 20 questions. The concept should be clear in your mind, because that is our priority. Our priority is to clear concepts with each and every questions. So that's why I give time to each and every question, so that I can tell you the background of that theory of behind that question as well, so that at least that concept, that topic which is related to that question becomes clear to you. I can solve 30 questions in one class, and I cannot explain anything about each and every question. I can just quickly move through questions, but that will not solve the purpose. My purpose is not to complete as many questions, but my purpose is to make you understand the topics. Okay? So first of all, you should know there are two terms which are being here used. First one is your primitive streak, and the other one is your Henson's note. Okay? So first term is, and your second term is Henson's note. So both the structures are formed in an embryo during gastrulation process. First thing is that both primitive streak and Henson's note, both the structures are formed during the gastrulation process. Henson's note is a group of cells, or a group of cells that constitute the organizer in the a, we are talking about the avian, so in the avian embryo. So now I will explain you in a little more detail. So, primitive streak is actually the first visible sign of gastrulation. The the option says gastrulation begin at the Henson's node. No, gastrulation does not begin at the Henson's node. Gastrulation begins at the primitive streak. Okay, Henson node is involved in later stages such as organization of the body axis, notochord formation. But the process of gastrulation itself starts at the primitive streak. So that means, what difference we can say, that primitive streak is the first visible sign of gastrulation. Okay, primitive streak is the first visible sign of gastrulation, and therefore it defines the your axis, your medial lateral axis and your rostrocaudal axis. All the axes are defined. It is the first visible sign of gastrulation. And Henson's note, at the anterior end of the primitive streak, there is a regional thickening of cells which is known as primitive knot or Henson's note. Okay, so at the interior end of the primitive streak, in the anterior end, at the anterior end of the primitive streak, you will have a regional thickening which is known as your primitive node or Henson's node. So what you should understand is that as the primitive streak, as this primitive streak elongates, the migrating epiblast cells, they join this primitive streak, and therefore they form the primitive node. And this primitive node, which is nothing but your Henson's node, it becomes the primary tissue organizer, where transcription factors and your chemical signaling, it drives the induction of tissue formation. So first is your primitive streak is formed, which is the first visible sign of gastrulation. And then as your primitive streak starts elongating, then your epiblast cells, they come and join, and therefore at the interior end of the primitive streak is your Henson's node formed. And this Henson node is responsible, it works as an organizer, where the transcription factors and other chemical signaling molecules, they drive the induction of tissue formation. So this is your first option. So your first option is true or false. It is false, because now we know that gastrulation, it does not begin at the Henson's node, it begins at the primitive streak. So this is your false statement. Then next is, hypoblast cells will form the future endoderm of the tissues. Is it true or false? No, it is false. Why it is false? See, there are two terms. First is your epiblast, and second one is your hypoblast. Epiblast is nothing but your primitive ectoderm, and hypoblast is nothing but your primitive endoderm. Okay, there are two terms. First is epiblast. This epiblast is nothing but your primitive ectoderm, and hypoblast is nothing but your primitive endoderm. Okay, so your epiblast, it forms your ectoderm, endoderm, and mesoderm, those layers. And your hypoblast, it forms the yolk sac, which give rise to chorion lately. So, does your hypoblast form the future endoderm? No. The ectoderm, endoderm, and mesoderm, those layers are formed by epiblast and not hypoblast. So, this becomes your wrong statement. Then next is mesodermal cells originate in the epiblast and migrate through the primitive streak to the developing embryo. So mesodermal cells, they are formed through epiblast. Yes, because we have already just now said that epiblast will give rise to ecto, endo, and meso, all the extra embryonic layer, and your hypoblast will give rise to yolk sac. Okay, it will give rise to yolk sac. So mesodermal cell originate in the epiblast, yes, and it migrate through the primitive streak to enter the developing embryo. So this is true statement. Then next is exogenous expression of nodal may result in conjoined twins. So yes, because nodal is a signaling molecule which is responsible for excess formation, your body excess formation. And abnormal expression can lead to the duplication of excess, therefore it can lead to conjoined twins. So nodal is responsible for body excess formation. Body axis formation. Then your last statement is the hypoblast is responsible for positioning of the gastrulation site. Yes, it is also true. Hypoblast, it basically plays a role in positioning of the primitive streak, which in turn determines where gastrulation will occur. So basically, hypoblast position your primitive streak, and when your primitive streak is positioned, then it will determine where your gastrulation site is there. So then your last three statements are correct. C, D, and E are your correct statement. Okay. I just a minute. I hope this is clear to you. Fine. Now we'll switch to go to the next question. These are lengthy questions. Actually, we just have three questions, but those three questions will take 1 hour because it's a very lengthy questions. Fine. Hmm. Which of the following statements are incorrect regarding mammalian axis formation? A. At the posterior end, brachyury inhibits Wnt3/8 that results in mesodermal cell formation. So, I don't know what is the exact pronunciation. Brachyury or brachyury, whatever you want to pronounce it at. Okay. Second is BMP4 induced Wnt3A is required for mesodermal cell formation. C. Anterior extension of nodal is induced by lefty 1 that promotes head structures. D. Caudin mediated inhibition of Wnt's is involved in anterior structure formation. And E is RA degrading enzymes at the anterior contribute to the RA gradient formation. So now you see why these questions take time. Now you just see all the options. Generally what happens, you will have MCQs where even if you have like 4 or 5 options, those 4 or 5 are options are related to the same topic. Okay, for example, you're talking about anyone, you're talking about the anterior nodal, for example, you're talking about BMP and Wnt pathway, interaction of BMP and Wnt pathway. So generally your four options would be related to BMP and Wnt pathway, but here you see each and every option are unrelated to each other. That means if you solve this question, your one, two, three, five topics or five concepts would get clear with this question. See, these all the options that are given here, these are unrelated options. This is about BMP and Wnt3 signaling. This is about RA degrading enzyme. This is about nodal signaling. So these are different. So just a minute. Hmm, what happened? I couldn't understand what happened. Is there any message? No? Okay. Okay, I don't know what happened. Fine. So if you solve this question, if you can answer this question, that at one time your five concepts will become clear. So try to solve this question. It's a very good question. Otherwise, anyways, I am here to help you. Okay, so I will give you 3 minutes. Right now it is 8:10. I will switch the screen at 8:13. Okay, try to solve it. One more minute left. Okay, the time is up. Hmm. Chalo, let's see. Hmm. Okay. So Amla has answered A and D are the incorrect answers. A and D. And Jasrina has answered B and E. So whether how okay, they are asking the incorrect. So A and D. Okay. And B and E. Hmm, okay. So see, these are the incorrect options. At the posterior end, A is incorrect. At the posterior end, Brachyury inhibits Wnt3A that results in mesodermal cell formation. BMP4 induced Wnt3A is required for mesodermal cell formation. Anterior extension of nodal is induced by lefty one that promotes head structures. And caudine mediated inhibition of Wnt is involved in anterior structure formation. These are the four incorrect statements according to the answer this. Now we'll try to analyze each and every statement. The first one is, at the posterior end, Brochure inhibits Wnt3A that results in mesodermal cell formation. So brachyury, what is brachyury? Basically, it is a transcription factor which is needed for posterior mesodermal formation. Okay, so brachyury is needed for posterior mesodermal formation and notochord development also. So therefore, will this statement be right or wrong? Here the statement says that it inhibits Wnt3A that results in mesodermal cell formation. Okay, one thing we know that it is required for mesodermal uh, s- uh, formation, but it does not inhibit Wnt3A. Rather, it has just the opposite role. That means it plays a role in promoting mesodermal formation by activating the Wnt signaling. So, it is written that brachyury inhibits the Wnt signaling, but actually it activates the Wnt signaling which is necessary for mesodermal cell formation. Okay, so this is an incorrect statement, and this should be activates. Okay, and in fact, there is a brachyury Wnt3A loop that allows mesodermal progenitor cells to remain viable. So these brachyury and Wnt3 they interact with each other, which helps in mesodermal cell differentiation. So then next is, BMP4 induced Wnt3A is required for mesodermal formation, cell formation. Now is it correct or wrong? It is again a wrong statement. Why? Just like we said that brachyury and Wnt3 work together. BMP and Wnt3A kind of work in an antagonist manner. Okay. In an antagonist manner, I'm trying to say, what does it mean? That both BMP and Wnt3 they are involved in mesodermal formation. BMP acts differently and Wnt acts differently. Okay, they're not completely antagonist. They are irrelated to each other. They're not related to each other. BMP and Wnt3A works parallelly for mesodermal cell formation. Okay, so BMP signaling acts alongside Wnt signaling, but it is not an inducer of Wnt. Could you understand? This is your Wnt BMP pathway, BMP4 for activating the mesodermal signaling, and this is the Wnt3A. They both promote, but there is no such interaction between them. So it is not like that BMP4 is inhibiting Wnt3 or BMP4 is inducing Wnt3. Nothing is happening. There is no relation between them. They are individually they are working. So therefore this becomes your wrong statement. Then anterior extension of nodal is induced by lefty one that promotes your head structure. First of all, what is the meaning of lefty one? What is lefty one? It is basically a protein that inhibits nodal signaling. Now here only I think the answer is there. Lefty one, it inhibits the nodal signaling. So here the question says, anterior extension of nodal is induced by lefty? No, because lefty does not induce nodal signaling. In fact, lefty inhibits the nodal signaling. Now how this lefty one protein inhibits the nodal signaling? It can do so by two ways. First one is that it can directly bind to nodal and it can inhibit the nodal signaling. This is the first way. Second one is it can bind to some of the core receptors of nodal signaling and it can inhibit. Are you able to follow? Because these are like very unrelated terms, I mean, unrelated concepts. And because we have time constraint, we have just 2 hours, so we cannot explain each and every topic in detail to you. So we expect that you have gone through the lectures of that week and you already have some basic knowledge, and therefore you are able to follow up. Even if you are unable to follow up, it's fine. You can just understand what I'm trying to say. You can go back, you can just read, go through the recorded lectures, and then again, if you're going through this PPT, then I think things will become more clear to you. Or if you have any specific doubts, you can just Google it and you can try to understand that specific concept. Then again, your topic would become clear to you. Because see, these are so many topics are covered in your 10 questions, 10 to 11 questions. We cannot explain each and every topic because it will take more than 2 hours and I have to cover the questions as. Therefore, I expect that you have at least a basic knowledge. But even if you are unable to follow any point, you can let me know. I'll explain that point again. Okay, if we, these are very unrelated terms. So if you couldn't follow any of the statement, just let me know. I'll explain it again. Okay. So yeah, we were this statement, yes. So lefty one, it actually inhibits your nodal signaling. Okay, and it do so by two ways. It can directly bind to the nodal, or it can bind to some of the co-receptors of nodal, and therefore it can inhibit. So from this we can conclude that lefty one inhibits nodal signaling rather than inducing it, and this is essential to limit the nodal activity to the posterior end. Okay, so therefore this is incorrect statement. I won't give you more details because then it will be really, it will go out of your head. Okay, fine. Your next statement is chordin mediated inhibition of winds is involved in anterior structure formation. So first of all, you should know what is chordin before understanding what is chordin mediated inhibition. So chordin is basically a protein which is involved in dorsal ventral patterning of embryo. I will write here. Chordin. Chordin, it is a protein which is involved in the development of dorsal ventral patterning of embryo. Now, chordin inhibits your BMP pathway. Another important thing is that cordon, it not only helps in your developing of the dorsal ventral polarity, but it also inhibits your BMP pathway by uh, inhibiting or stopping the BMP from binding to its co-receptors. So the question here says that chordin mediated inhibition of Wnt is involved in anterior structure formation. So, is it a correct statement or wrong? Wrong. Because chordin inhibits BMP signaling, not the Wnt signaling. Here it is written that chordin inhibits Wnt signaling. No, chordin does not inhibit Wnt signaling, it inhibits BMP signaling. Okay? So, cordon is crucial for hair development, that is fine, but it does so by inhibiting the BMP pathway and not the Wnt pathway. So, this statement again becomes incorrect. Yes. Then, RA degrading enzymes at the anterior contribute to the RA gradient formation. What is RA? RA stands for retinoic acid. So, retinoic acid degrading enzymes, one such example of retinoic acid degrading enzyme, which is a highly studied example, is your CYP26. CYP26. It is an retinoic degrading enzyme. So, they degrade your retinoic acid at the anterior end, which helps in establishing a proper gradient disseminate, which helps in the establishment of a proper gradient and axis formation. So this statement becomes your correct statement. Hmm. So from this we can say that the first four statements are incorrect one. The last one is correct. I am just repeating it again. The first statement says at the posterior end, broccoli inhibit Wnt3a that results in mesodermal cell formation. So no, it does not inhibits the Wnt3a, it actually activates it. Then BMP4 induced Wnt3a is required for mesodermal cell formation. So although BMP and Wnt3a both are required for mesodermal cell formation, but they both work individually, parallelly. There is no intersection between the two pathways. In this case, anterior extension of nodal is induced by lefty one that produce head structure. So lefty one does not induce nodal, it actually inhibits the nodal signaling. Then chordin mediated inhibition of winds is involved in anterior structure formation. So chordin, it is basically a protein which is involved in dorsal ventral patterning of the embryo. And yes, it is true that chordin is involved in anterior structure formation, but it accomplishes its task not by inhibiting the wind pathway but the BMP pathway. Then RA degrading enzymes at the anterior end contribute to RA gradient formation. RA means retinoic acid. So yes, it is a correct statement. So retinoic acid degrading enzyme, for example, CYP26, it basically degrades the retinoic acid in the anterior end, which helps in establishing a proper gradient for the axis formation. Okay. So I hope this question is clear to you. Now we'll move to the next question. So generally what happens is that we start with simple questions and then we end with a little more trickier questions. So uh, now we have this question. Yeah, it's a very confusing question. So it can have many this thing um, reasons and many I mean you can have many ways in which you can analyze this question. You might not align with the answers also, but that's fine. We'll try to understand. Our basic objective here is to understand the concept. Okay, then we'll see whether the answers that are given to us are actually correct or not, but at least we should understand the concept which they're trying to ask in this question. Okay, so the question says a mutated embryo was dissected to isolate the prospective anterior half. Okay. The following plot depicts the protein levels in the prospective anterior half. Based on your understanding of the interactions involved in mammalian axis formation, identify the correct inferences for the following plot. Please note that in the plot, uh, where it is, yeah, this is your chordin, this is your Dikpoff, this is your noggin, this is your wind, and this is your BMP. This is your ladder control. So the options are the cells may have a gain of function mutation in BMPX. The cells may have a gain of function mutation in WNT-X. The extension of BMPX into the anterior half is due to the loss of function mutation in the chordin. Exogenous application of chordin may rescue the altered development, if any. So I'll give you 4 minutes because it's logical questions and you have just dealt with a very uh, big conceptual question. So 4 minutes, think and try to solve this question. First of all, when you have such questions, so if you have attended the previous lectures or at least you have solved the assignments, you must have an idea now that sir gives some data, I mean these kinds of visual data type questions. So that means in your final exam, for sure one question of this kind might come, and these are actually very good questions because they help in clearing your concepts. These are not like you just have to mug up and write. And also these are the questions that comes in your competitive examinations also. So if you can solve these questions, that means your concept which is behind this question is pretty clear. So therefore I'm giving you 4 minutes. It's 8:28 right now. So try to solve this question. Try to understand the question first. If you are able to understand the question, half of the thing is already done. And then if you have a basic understanding of this, all the molecules and their pathways, then I think it will be easy for you to answer this question. So time starts now. It's 8:29 now. Okay, it's fine. 8:33. I'll switch the screen. See all the options, and also you should know that this blot is what kind of blot it is given. It's a protein levels. So what kind of blot it is? 3 more minutes left. So the horizontal lines that you see in the blot, it represents its expression level. It's a hint. So those expression levels can be correlated with the options given. For example, in cordon and BMP. See the difference in the thickening. The thickening represents the expression. Thicker the band, more is the expression of that protein. Thinner the band, less is the expression of that protein. Okay. Now I try to solve it. 1 minute left. Okay, now I'm switching the screen. I hope you have tried to solve it. Hmm. Okay, let us see. Okay, Amla has answered A and C. Uh, and then he has also answered A, C, and D, you are saying, okay? A, C, and D, you're saying. Okay, okay, fine. So let us try to understand this. So, first of all, this is a which kind of blot is it? So, this blot depicts the protein level. That means it is a what kind of blot is it? It is a eastern, western, which blot it is? Is it a northern blot? What kind of blot it is? It is a western blot. Good. So it is a western blot. So western blot represents the protein levels. So this is, if you see, this is the ladder. Okay, this is the ladder. So this ladder represents the any known protein whose size is already known. So we run that ladder so that we can compare the whatever in test samples, whatever expression we got, we can compare it. For example, um, yeah. So let us take this only BMPX. So you know that if this is, let's say it is, I don't know, if it is 20 kilodalton, so you can say that it is somewhere between 10 to 20 kilodalton. The exact, the all the sizes are not given. It's just like in so from here you can get a little estimation that okay, whatever protein that you have got from this lane, it is something in between 10 to 35 kilodalton, or it is 10 to 20 kilodalton. So this is a ladder which acts as a reference point, a positive control also you can say. So always whenever you are doing any kind of electrophoresis and you run gels, um, for example, you are doing agarose or page polyacrylamide, always whenever you run gel, you have to plot or you have to load the ladder, because that ladder will only tell you the estimate or the approximate band that you have got. Fine. Now, the plot in the image, it shows the protein levels of several of the key proteins which are involved in axis formation. Okay, the question says that an embryo was mutated, and then that mutated embryo was dissected, and then we have the anterior half of the embryo. So this blot actually represents the protein level of that prospective, means that would be anterior half, and based on the understanding of the interactions which are involved in mammalian axis formation, we have to tell. So first is okay, the correct answers are: the cells may have a gain of function mutation in BMPX, the cells may have a gain of function mutation in WNT-X. That means A and B are your correct answers according to this question, according to the one which was given. Okay, so the cells may have a gain of function mutation in BMPX. So for solving this question, you should first know what is the function of these individual molecules. So cordon and BMP signaling are opposite to each other. So cordon antagonize the BMP signaling. So I'm writing here cordon and BMP. These are antagonists of each other. So if cordon in so cordon basically is involved in anterior formation, anterior development. So cordon is involved in anterior development. What does it mean? It means, see, the embryo that has been dissected, we are seeing the anterior part of it. So in the anterior part, cordon should be expressed, because cordon is responsible for anterior development. And since cordon would be expressed, what will happen to the BMP level? BMP levels will go down. So BMP has to go down at the anterior level, and cordon has to be upregulated in the anterior level so that proper anterior formation takes place. So in a normal case, in a normal embryo, this is a mutated embryo, in a normal embryo we would expect higher levels of cordon because the anterior region is rich in cordon and cordon suppresses BMP. Okay, but in this case, since it is a mutated embryo, so there has been some mutation. So in that mutation you can see that BMP levels are a lot higher here. Okay, that means there is some gain of function mutation in the BMP that has led to more expression of BMP even in the anterior region, and cordon levels are very less. So in a normal case, the expression of BMP in the anterior part should be less, but there this is an mutated embryo. So there has been some gain of function mutation. Gain of function means that BMP has gained its function. Now it has become even more prominent, and therefore you can see the band is so thicker. It should be a thinner band or very not at all. So therefore A becomes the correct answer. Second is the cells may have a gain of function mutation in the Wnt-X. Now, Wnt, DKK, in a normal case, what should happen? Is this, yeah. Uh, I don't know where the cursor goes. Yeah. In a normal embryo, DKK would suppress the Wnt, and DKK is responsible in the anterior half. So, DKK is a Wnt antagonist, and DKK is crucial for head development, and Wnt has to be suppressed in the anterior side. So both BMP signaling and Wnt signaling has to be suppressed in the anterior part. BMP is suppressed by cordon, Wnt is suppressed by DKK. Okay, but here you can see that there are higher levels of Wnt. Okay, there is higher level of Wnt and there is higher level of DKK also. That means DKK is there and DKK is trying to inhibit Wnt, but Wnt is present in more than the required concentration. So there has been a gain of function mutation in the Wnt. That is why even DKK is trying to inhibit it, it is not getting inhibited because it is more than DKK here. It is more than high, very much higher than the required amount. So there has been a gain of function mutation in Wnt. Otherwise, if DKK is there, Wnt should be less. But since there is a gain of function mutation, now it is out of the control of DKK. So even if DKK is present, still the protein band of Wnt is very much high because there has been a gain of function mutation in the Wnt pathway. Fine. Next, the extension of BMP-X into the anterior half is due to the loss of function mutation in cordon. Now, see, you can say that BMP is highly elevated and cordon is less. So, by seeing BMP, we can say that there has been a gain of function mutation. But we cannot conclude that BMP is more because there has been a loss of function in cordon. See, if we say this, we cannot say this. This is more probable because you can see that BMP band is more. Now, the more expression of BMP in the anterior portion, although BMP should not be expressed in normal case, because in normal case BMP is inhibited by cordon. But since here is a probability that there is a gain of function mutation in BMP, but it doesn't means that there would be a loss of function mutation in cordon. No, it, see, there can be a case that since BMP is had a gain of function mutation, so it is inhibiting cordon, but it does not necessarily mean that there is a loss of function mutation in cordon. Therefore this becomes wrong. Likewise, exogenous application of cordon may rescue the altered development. Can it rescue? No. Why it cannot rescue? just because why DKK could not inhibit Vintex. You can see here, even though there is high expression of DKK, even though it is unable to inhibit Vintex because there has been a mutation in Vintex in such a way that now it is under uncontrolled development. So similarly, even if you are giving cordin exogenously, it cannot help in inhibiting BMP because now BMP has been mutated. It will have uncontrolled proliferation of BMP. So this also becomes wrong in this case. So because there has been a gain of function mutation in BMPX, therefore, even if we provide cordin from the exterior exogenously, it cannot inhibit BMP just for the same case as DKK could not inhibit Vintex. Okay, is this question clear to you or not? If you have any doubt, you can ask me. Was this, could you understand? Could you follow? If you have any problem, you can ask me because it's a conceptual question. I can understand if you have any queries. Was this question clear? Okay, fine. So I hope you could understand this question. Always remember that if you have a gain of function mutation in any gene, that means now even if the gene which is generally involved in suppressing that gene, it won't function because it has now a mutation, a mutated gene. Now it becomes uncontrolled. Fine. Now, next question. So, this is the last question for the day. We'll see if I can explain this question in 15 minutes. If not, I'll try to explain it and I can explain it again in the next class so that it becomes clear because these kinds of questions do take time. I understand. Okay. So, the question says, three pregnant females were taking a newly launched drug for morning sickness. Okay. After birth, their children were found to have defects in their vertebral column. Fine. Given below are some of the statements made regarding these drugs and their interactions with Hox genes in 3' to 5' order: Hox A, Hox B, Hox C, Hox T. Select the correct inferences. A. Drug A may have caused gain of function mutation in Hox B. B. Drug A may have caused gain of function mutation in Hox D. C. Drug A may have caused a loss of function mutation in Hox C. D. Drug B may have caused a loss of function mutation in Hox B. E. Drug B may have caused a gain of function mutation in Hox D. And last, drug B may have caused a gain of function mutation in Hox A. So I'll give you 3 minutes. Try to understand this question. See, it's 8:47 right now. I'll switch the screen at 8:50. I'll try to explain this question in 10 minutes. If not, I'll again explain in the next class. So don't worry about that. Your objective should be to understand the concept. Okay? Now you try to solve. If you know the function of Hox genes, these Hox A, B, C, D, then it is very easy for you. And you should also know how to analyze these kinds of graphs. This is the control. So you have to analyze with respect to your control. What are the levels with respect to control in drug A? What are the levels with respect to control in drug B? Okay. So now try to solve it. Two minutes left. 1 more minute left. Hmm. Time's up. So Amla has answered C, D, and E. Okay. The correct answers. The correct answer is okay. Drug A may have caused a gain of function mutation in HOXB. Drug A may have caused a gain of function mutation in HOXB. Drug B may have caused a gain of function mutation in HOXB. And drug B may have caused a gain of function mutation in HOXA. Okay. So see here, what they are trying to ask in this question. So this is the data which shows the effect of drug A and drug B on the vertebral column. Basically, how these drugs are impacting the development of different vertebra. That is, the blue one represents the cervical region. This represents the orange one represents the thoracic region. This is the lumbar region. This is the sacral region. And so basically, there has been a mutation in the HOX gene. So if you know what are HOX genes, so HOX are the homeotic selector genes which are responsible for your um mm, pat homeotic genes. Basically, these are responsible for your anterior posterior axis. So now important are the most crux of this question is that HOX A and HOX B. So what I'll do it here, I'll take this, okay, and the options. Sorry. Yeah. Okay. This is your question. Now you see HOX A and HOX B. HOX A and HOX B. They are responsible for anterior structures. Okay. They are responsible for anterior structures, which are your cervical and thoracic. Okay. Like cervical and thoracic. Your HOX C and HOX D. They are involved for the patterning of your posterior structure, which is your lumbar and sacral. Okay, so this is important. This will be, if you know this, you can solve the question. This is important. Now you see here, this is the control. With respect to control, you can see that in drug A, this particular area is increased, which is the thoracic region. That means thoracic region is increased over here. Can you see it? With respect to control, the thoracic region has been increased. The cervical region is somewhat decreased over here. So, first option says that drug A may have caused a gain of function mutation in HOX-B. So, is it true or false? It is true. Why it is true? See, HOX-B is responsible for the anterior structure formation, which is cervical and thorax. You can see that there is more thorax as compared to control. That means it might be a possibility that HOX-B has gained a mutation, like a gain of function mutation has been there in HOX-B, and because of that, the thoracic region has increased. So this looks like a correct answer to me. Then second statement is drug A may have caused a gain of function mutation in HOX-D. So will that be a correct statement or an incorrect statement? Hmm? It can also be a correct statement. Why? Because you can see that there can be a gain of function mutation in HOX-B and there can be a gain of function mutation in HOX-B. Why? HOX-B, you see, this thoracic region has increased. HOX-D is responsible for posterior structures such as lumbar and sacral. You see, here the sacral portion is more here. Can you see? Where is the cursor? See, although this has been decreased, but this has increased. So there can be a possibility that HOX-D can have caused a gain of function mutation in HOX-D which is responsible for posterior structure formation. Then drug A may have caused a loss of function mutation in HOX-C. HOX-C, loss of function mutation in HOX-C, we cannot say because this particular region has increased. So we cannot say that it has a loss of function mutation. Then drug B may have caused a gain of function mutation in HOX-B. See drug B, HOX-B. HOX-B is responsible for anterior structure formation. You see, this is the cervical region. So, with respect to control, the cervical region is more. So, there can be a case that there is a gain of function mutation in HOX-B. Therefore, this particular region has increased. So, this can be the case. Drug B may have caused a gain of function mutation in HOX-D. HOX-D stands for this thing. Okay. So according to the answers, they are saying this is a wrong because C drug B is there and HOX-D is responsible for posterior structure formation, lumbar and sacral. So you can say that I don't know why they have mentioned E as the incorrect one. E, because still there is a little bit increase only. But chalo theek hai, we'll keep it as wrong. We'll see the last one. That is, drug B may have caused a gain of function mutation in HOX-A. So HOX-A, yes. In HOX-B or HOX-A, there can be a gain of function mutation because the cervical portion has increased. Compared to this, you can say that there cannot be a gain of function mutation in HOX-D because D is responsible for posterior structure. This is same, and this is same with respect to control. So your sacral is same with respect to control. Little bit increase is there, but is not that much prominent probably. That's why they have marked it as wrong. The concept behind this question is that you should understand the function of HOX-A and HOX-B, HOX-C and HOX-D genes. HOX-A and HOX-B are responsible for anterior structure patterning. The anterior structures include your cervical and thoracic. Hox C and Hox D is responsible for the patterning of the posterior structures, which essentially are your lumbar and sacrum. And these are the levels that should be in a control, or in a control, I mean in the normal case. In a normal case, these should be the levels of all the vertebrae. Okay, here you can see that these drugs have caused in decreased, I mean change in the expression of the cervical, thoracic, lumbar and sacral. So then we have to interpret it and analyze it with respect to the function of Hox genes. That here you can see that this particular portion is highly enhanced, the thoracic region is highly enhanced. So there can be a gain of function mutation in HOXP, because HOXP is responsible for anterior structure formation, which is cervical and thoracic. So like this, you have to try to relate it with the function and the graph. So I hope I was able to make you understand. One very important thing which I wanted to say, that whenever you come for learning, whenever you are attending any class, just leave my class, any class in general, when you are attending, there is something that we need to learn. Also, more important, learning is unlearning. That means you don't have to only learn things, but you have to unlearn the wrong concepts also. So your mind should be flexible enough to unlearn the wrong concepts that you might have had, or some flexibility should be there. You should be accepting to the new concepts and to the new logical explanations. Okay? This is I wanted to say, that whenever you are going in any class, it's not only about learning, it's also about unlearning. Okay? So now we'll meet in the next class, and I hope something you were able to understand something from this class. So we will have two more classes. The next class will be the course completing class, and then we'll have a revision class afterwards, where we will try to summarize everything. So thank you. Have a nice week, all of you.